Okay, I think it's all good.
Okay, so uh, I'm going to be talking a bit about the prediction of genetic informations like mutations or gene expression from digital pathology. The first work I did in this is shown here on top, which is the prediction of TP53 mutations in prostate cancer from digital pathology. That was kind of the first work I did. And then more recently, we published the work below, which is the prediction of gene expression, also from digital pathology, which is more pan-cancer, so 16 cancer types. And I'll be more focusing on the second one here. And maybe before I go into details, I just want to say a little bit about why we are going through this. And that's because in current like oncology clinical practice, the information that doctors have is quite limited. So they will usually have the age, grade, and so on of the tumor, and pathologists will visually examine the tissue slide to do diagnosis and prognosis. But it's kind of hard to, based on that information alone, say like, what are the chances that a tumor might metastasize or what the optimal treatment for a patient may be. And so to do that, researchers have been looking into finding genetic biomarkers that can give a better indication about metastasis or therapy. But obtaining these genetic biomarkers is super expensive. For bulk sequencing and especially for spatial sequencing, it's way too expensive to implement it into clinical practice. So that's why we are developing these models to predict them from pathology. And the main problem here is tumor heterogeneity. So over the course that a tumor develops, there may be different subpopulations that have different mutations. And because sequencing is so expensive, you cannot test all different regions in a tumor. And you may test a certain lesion on certain mutations or gene expression, but you may be testing the wrong one. Like there may be a more aggressive one somewhere else that you didn't test, and you may underestimate the aggressiveness of the tumor. And this heterogeneity is, of course, also reflected in the whole slide image. So if you have the image on the left, we had a pathologist annotate different regions in the tumor. And as you can see in these zoomed in versions from each region, they visually look very different. So they look visually different, but of course, they can also have a different molecular profile, so different genetic mutations or gene expressions in each region. The goal of our models is to make a spatially resolved prediction of these genetic mutations or gene expression, where you would have this heat map that shows a low or high predicted probability of a mutation being present or a gene being high or low expressed. So how do we train such a model? Of course, in deep learning, we need matched image and outcome pairs. But the problem is here that you have this tumor heterogeneity. So where in your image may a label be valid? And in one case, you may have a label for each tile. This would be the case, for example, if you have spatial transcriptomics data where they really have for each specific tile that they test the genetic sequencing of that tile. In the other case, which is way more prevalent in digital pathology, you have a label that is only available on patient level. So this would be if you want to predict metastasis directly from the image, you don't know upfront, like where is that label valid? Or in bulk sequencing, you also wouldn't know where in the tumor it was sequenced. So in the first option, is the easiest one. So there you would have your image outcome pairs for each tile. This gives you very good performance and very good training because you may have hundreds or sometimes thousands of tiles per your whole slide image. But the downside is that this is very expensive or impossible in some cases, like if you have only metastasis as label for your entire image. But if you do have these image outcome pairs per tile, how it works is you have this, for example, annotated region here on the right, the red region, where you extract tiles from that region and you know that your label is valid here. So you will train a separate model for predicting in each tile if your property is present or not. And this is kind of what we did in my first paper for the TP53 mutations. And we compared what if you use this very specifically annotated region for training versus if you are more uncertain. So if you would use all tumor tissue versus the entire image. And we saw very clear like drop in performance the more uncertain you are about your region where the label is valid. So what if you don't have these tile level labels? So in that case, you have noisier training, and it also means that you need larger data sets to achieve your training convergence. The advantage, of course, is that it's cheap and always applicable. In this case, you would have your tiles in the input, where a first deep learning model is going to extract features for each tiles. And then in the most simple setting, you would take a simple average of your features to make a prediction. But if you do the simple average, you may mask out some small signals in your input. So maybe there's like one or two tiles that are very important for your end prediction, and this will lose that signal. So recently there's research being developed where this average is replaced by a second deep learning model, which is going to learn which tiles in your input are relevant for your prediction. Of course, this introduces more parameters and so more chances of overfitting. So now that I gave like this small introduction, I can go over to the specifics of our SEQUOIA architecture, where we have the slide level training. So we have all tiles in the input, and then we benchmark two different feature extractors. So the traditional ResNet feature extractor that's pre-trained on ImageNet, which was used for a long time, up until recently, where you had foundation models that were being released specifically for whole slide images, like the Uni foundation model. And then you have these features, so 1 to n, which are extracted by the feature extractor. Whole slide images have a very, very large number of tiles, so there can be like thousands of tiles. And that's too much for us to build upon later with the deep learning model, because like complexity explodes, and there's also quite a lot of redundance here, because like tiles that are very close to each other may look kind of similar. So there may be a way to compress that information and make the input dimension a bit smaller. So we chose here to apply k-means clustering, where we are going to take together these tiles that have pretty similar features and compress them into 100 smaller clusters. And then within each cluster, we just take the average of the features. Once we have these 100 features, we have the second deep learning model that I mentioned earlier that is going to be the aggregation, and that is going to then predict the gene expressions, which is the output of our model. About this aggregation, so the first one is MLP aggregation, which was introduced in HE2RNA. And maybe I can show this easier here. So what happens is you have the first tile, like super tile vector, they call it after clustering. And each of these goes individually through the MLP. So each tile is independently transformed. And after this transformation, HE2RNA does like a weighted average to make the prediction. But this kind of approach doesn't focus on any context. So to determine whether a tile is relevant for the prediction, you only focus on that tile. While it may be the case that a specific tile in combination with a certain other tile becomes relevant. So this is what is being modeled with transformer aggregation, which was introduced in tRNA's former. So as you may know, transformers, they use self-attention modules, which you can see here on the right. And very simplistically explained, if you have XT on the input, which is your tile, it will look at all pairwise combinations of tiles. So it will compare each tile to each other tile to have a context-aware representation. But this is quadratic in complexity and therefore overfits quite easily if you don't have a lot of data. So in our model in SEQUOIA, we used the linearized version of the transformer, which was introduced somewhere early last year in speech recognition. And the module you can see is here on the right, which is a simplification somehow of the self-attention module, where each tile in the input xt is not compared to each other tile anymore, but it's compared to the sum of vectors in the input. So the sum is, of course, linear instead of the quadratic component we had before. Okay, so now we have the model. Just a quick few words about how we evaluate it. So if we have the predicted gene expression for our 20,000 genes, and we have the ground truth values for all of those genes, we have three kind of metrics that we look at. The first one is Pearson correlation for a specific gene across patients between the predicted version and the ground truth. Then we have the root mean squared error. And then we also define a set of statistical tests where we compare the output of our trained model versus if we would just use a randomly initialized untrained model to say like what is here significantly predicted and what is not. We use three cohorts for the bulk evaluation in our model. We have TCGA that we use for model development, of course, split and train validation test. And then we have two independent cohorts, CPTAC and Tempus, that we use to evaluate the generalizability of the model. So this is the performance on TCGA, and it's kind of a big figure, but I'll break it down very quickly. So on the x-axis, we have different cancer types. Then you see some violent plots, which represent distributions of the left axis, which is correlation coefficient. So this is for the top 1,000 genes. What is the distribution of correlation coefficient between prediction and ground truth? And we have 6 violent plots representing 6 different models where we benchmark the variations that I talked about earlier. So ResNet feature extraction versus uni feature extraction. And then we have 3 aggregation schemes, MLP, transformer, and the linearized transformer. The rightmost one, the dark yellow one, is SEQUOIA with the linearized transformer and uni model, which across all cancer types is consistently the highest distribution plot, so with best correlation performance. And then on the right-hand axis, we have the number of genes that can be significantly predicted with the model, which is indicated with these squares. And for example, you can see the best model here for breast cancer. The SEQUOIA model can predict a bit above 17,000 genes with statistical significance. About this performance, we saw that, which is kind of logical, that for achieving best performance, we have cancers with the most available slides. And then we have worse performance if there's not enough data available to train the model. And this is pretty consistent across different models. So in this plot, you see different models in different colors, and each dot represents a specific cancer type. So we plot the number of slides per cancer type and then the number of significant genes we have with the model. And you can see that across all models, there's like a pretty positive correlation there. This also translates to the generalization on CPTAC. So we still have that breast cancer is the best performing model with pretty good generalization capabilities of SEQUOIA. So then in the next step, we have looked at, okay, so we predict all these genes, but are they useful? Like, what do they capture? And for this, we performed gene set variation analysis, both across cancers, and then we looked also to cancer-specific processes. And the across cancers performance is shown here on the left, where you have a matrix that shows the number of genes in a specific pathway that is present. So 270 genes in this cell cycle regulation one for, I think this is liver cancer. And then the color represents the log p value of the gene set variation analysis. And we see that across cancers, there's several like cancer-related processes that are present, like cell cycle, cell matrix adhesion, and so on. And then also for cancer-specific processes, we have like dendritic cells and stuff like that. So we have some analysis that shows that the genes we capture are really useful. And now, so that we have this gene expression prediction from the digital pathology, we want to see how we can use this downstream for a clinical prediction. And we benchmark this with the other setting where you would use a deep learning model to directly predict the clinical prediction from the histology image. So here you have for breast cancer, we took a gene expression signature, which is shown here on the left, like some genes, not all of them. I think it's 270 genes in the signature. And we built a kind of regression model to predict recurrence-free survival for the breast cancer patients. And in the middle, you see that if we use this predicted gene expression, you can stratify between low versus high risk patients in this group. But if you would use histology directly to predict the same thing, then it's much harder to make the delineation between low versus high risk groups. And then in the last part of our model, we want to try to predict spatially resolved gene expression. So until now, we did bulk prediction, and we need to find a way to transform this bulk prediction into a spatially resolved method. And for this, we did a kind of hack in the model. So we know that we have 100 feature vectors in the input. And what we did was we transformed this into a 10 by 10 input window. I mean, I show 4 by 4 here, but imagine it's 10 by 10. What we do then is we make the prediction for if we use all these tiles as input, and then we save the prediction for all tiles in the window. Then we move one tile to the right, we save all the predictions again, and we repeat until the window is covered the entire image. And then for each of the tiles, we average the outputs across all windows. And this gives us a spatial prediction. We have also a way to evaluate our spatial prediction because we had access to a spatial transcriptomics data set of 18 GBM patients. And what you see here is for this whole slide image on the left, we have a certain prediction of a certain gene here, the HLA-DP-B1 gene. And then we also have the ground truth on the right. We have two evaluation metrics, a pixel-wise evaluation metric, which is just a Pearson correlation if you flatten both the prediction and the ground truth. But this pixel-wise metric is not always ideal because it's pixel-wise. So it doesn't take into account, for example, whether your prediction may be shifted by 1 pixel to the right or left. Then Pearson correlation might reflect poor performance. So that's why we have also this 2D Earth movers distance as evaluation metric, where you are going to take into account like how much has your ground truth shifted or your prediction shifted. So we did this across a whole set of genes and all 18 slides, and we found that there are a good number of cases where we have a pretty good spatial prediction. Then the next thing what we did is when we have these set of genes, we wanted to look like which genes are co-located. So which genes show high expression in the same areas of the slide. And we made this kind of matrix that has for each pair of genes, whether they have a high spatial correlation or not. And then we clustered this matrix And we saw what emerged was that genes that have or that are representative of a similar cell type, that they co-occur together in the image, which is biologically meaningful. And this was one of the ways that we were like, okay, our model is doing something that makes sense. And once you have this prediction, this allows you to also then visualize on the image, like, okay, this group of cells is probably from this cell type, and this group of cells is probably from that cell type. So that was kind of the paper summarized. So we have this cost-efficient prediction of gene expression from the whole slide images. We use that to make a downstream clinical prediction, and we are like on the way to finding spatially resolved predictions of gene expression. So thank you for your attention, and let me know if there's any questions.
That was really great. Thank you so much for presenting such a cool project. Yeah, so I think I will let maybe anyone else have any questions. Go ahead. Vidya. Shall I maybe read it out loud, or Vidya, do you want to ask it over the mic if you don't mind?
Hi, Maria. That was a great presentation. Thank you so much. I had a question regarding the clustering technique that you used, which was k-means. How did you exactly arrive at k is equal to 100? I mean, did you play around with other numbers as well? So yeah.
Yeah, so very good question. We did a very quick benchmarking in the very early stages of our project. We tried like, I think it was 50, 100, and maybe 500. And then we just decided to go with 100. But ideally, of course, you would test out more different combinations. But we just didn't have the time to thoroughly benchmark that, but we just went with 100.
Okay, thank you.
Okay. Anyone else got any questions? No, I've got quite a few questions, so maybe I will go ahead. Yeah, so I think it's a incredibly important project, and it seems quite timely as well. With so many sort of data sets being released now for spatial transcriptomics, I think it was a really cool time to be working in this field. So one question I sort of have is, could you maybe have evaluated your model against the HEST 1K dataset from the Faisal Mahmood's lab? Because I think it's like 1,000 spatial transcriptomic slides. So maybe that could be sort of something that's validated to sort of see how well your model performs on a sort of mass scale data set.
Yeah, that's a very good point. So I think the breast, so we did a GBM spatial transcriptomics evaluation, but we also had a breast one. And I think that one is from the HEST 1K. But I do have to say, like, the publicly available spatial transcriptomic studies, they usually have pretty bad quality H&E. So we do see that I think that performance that we are evaluating on these publicly available data sets is like inferior to what it could be if we had much better quality H&E slides available. And so that I think is the main downside of currently spatial transcriptomics data sets. It would be super cool if we had some with better H&E quality to really evaluate what these models can do spatially.
Yeah, because I think with most spatial transcriptomics tools nowadays, it's like you do the sort of the spatial transcriptomics first, and then with the same slide, you can then do H&E afterwards. So I think it's usually the afterthought of the study as opposed to the sort of pre-thought, which is a shame because I think if you can have really good quality data on both aspects, you have quite a lot of power in that aspect. And then yeah, I was going to then ask. I got two more questions maybe. So yeah, instead of like the k-means for sort of, I guess, because there probably is redundancy in terms of like there's a lot of slides that will have, or patches that will have the same sort of underlying morphology. But did you sort of benchmark if the addition or removal of this k-means before the aggregator has a much of a difference? Because you may find it like it may be limiting as a bottleneck of information, or so allowing all the features to pass through to the to the aggregator, they may have some benefit. I'm not sure. Did you see any differences in that?
So we actually didn't benchmark that
Mhm.
because we had some papers and we looked into input sequence length that are usually used for transformers and stuff, and I think they are usually limited around 1,000. So if you have 10,000 tiles for your H&E, then
Mhm.
I don't know if the transformer is going to work or how that would turn out. But it could be maybe interesting to just try and see what comes out of that. But we didn't do it. No.
Yeah, because I guess
And
sorry, go ahead.
oh, yeah, I had a question. Do you think there would be an interest in using an iterative approach where you first identify the different domains in your image and then kind of reiterate on that by focusing on domains only in like a two-stepped approach, two-step approach?
Yeah. You mean like during training that you then kind of
Like
find a way?
you train a first model to identify the domains, and then you train the final model, which will predict the gene expression by focusing on those identified domains.
Yeah, I think that could be an interesting approach. But the only thing I'm kind of thinking about then is how do you where is your signal for interesting domains? Or would you train it like end to end? Maybe that could be an option. Yeah.
Yeah. No, I guess I also had in mind just domain segmentation from spatial transcriptomics, but I guess that's not
Mm.
necessarily an option here. Um, but I'm not familiar too much with the field, but I would guess that there are methods which just do domain segmentation without doing anything else on purely H&A slides, no?
Yeah, I think for prostate cancer, you could maybe do like Gleason grade prediction, and then you would have different Gleason grades in your image, and maybe there could be a way to then just use that. Yeah, I think the I think now that like feature extraction is kind of solved with the foundation models, I think the most value will come from this, like finding a good way to identify which tiles to use and how to aggregate them. Yeah, definitely.
Thank you very much.
Yeah, and I was gonna then ask. Well, I guess it's more of a commonplace. Sort of would be good to hear your opinion about it as well. Is I guess for specific sort of transcriptomic signals that you're not very well, the model is not very good at predicting whether or not that's as a result of the model or just the fact that those transcriptomic signals don't present morphologically or there's no sort of pathology to it. So I get using this kind of approach in the first instance is a sort of good way of finding out whether or not like a specific signals are presenting physically or not, because it could just be almost like it doesn't really have much of an influence in terms of sort of the actual morphology of the H&E or the environment within the cancer. So I think this is sort of an interesting approach for sure. It could be sort of to see which signals are important or not in that aspect. But yeah, I think
Yeah.
yeah, go ahead. Sorry.
No, I just wanted to say, like in my first paper where we predicted TP53 mutations, we saw that like the model is probably not predicting the real mutation, but it's more predicting like the effect the mutation has, and that effect may also be achieved by other alterations. And we saw that the model makes like a lot of false positive predictions. And in those false positive patients, there were often like deletions in TP53 or like some other mutation that may trigger like the same downstream effect. So yeah, definitely. I think we still need to see like what the biological limits are. Like, can we predict genes, mutations? Expression is easier, I think, or better related to the H&E image than a mutation. So I think that's why we moved to gene expression instead of mutations. But yeah, there as well, still a lot to think about and to analyze.
Yeah, great. And I guess one more last one, I think for me. Um, yeah, like how can you sort of imagine this approach in general, not just your model specifically, but future iterations as well, being implemented in sort of like clinical diagnostic workflows? Because I think there's probably quite a bit of power in that, because a lot of health systems across the world are very stretched financially. So, if we could sort of resolve this back to just using H&E, H&E alone, which is quite cheap by comparison, it could be quite a good approach potentially. But yeah, it would be good to sort of hear your thoughts on that.
Yeah, it would. I think it would be really, really great if we can like avoid this expensive gene expression sequencing and so on. But I think there we do need like a lot of evaluation still and a lot of. I think we need. The problem is that we don't have enough like diverse training sets yet. I think it has become better with the foundation feature extraction models, but we still see a kind of big drop if you evaluate it on other data sets. So I think, yeah, it's very promising, but I'm always kind of I'm not sure if we're there yet, but yeah.
Yeah. No, it's good to have a
And
little
so
bit of skepticism. Sorry, go ahead.
Sorry, in that line of questioning, what do you think are the missing ingredients to bring the accuracy of this kind of model to be high enough so that it can be used in the clinic?
Yeah, I think it's a very difficult thing to to like predict because obviously for sure more data will be necessary. And I think with the uni models and stuff, we see such a big improvement that it makes sense to believe that this will help. But as we saw, like in the LLMs and so on, maybe the added value of all that bigger pre-training and stuff may not be enough. So I don't know. I think this is a very like unique setting where if you want to predict these genes, you have the added complexity of is it even biologically possible? Like, do we have a threshold until where it's feasible to predict, or can we just make it better with more data? So yeah, we'll
Great,
see.
thank you very much.
Great. I think if there are no more questions, I'd like to, thank you so much for spending the time on, making the presentation, this amazing project. And yeah, thank you very much for sharing it with us.
No, of course. Thank you for attending, and it was nice talking to you all.
Great. Thank you very much, everyone. Now I'll go ahead and end the meeting.
Thank you. Bye bye.